Sarah Gilbert, Vaxxers, the inside story of the Oxford AstraZeneca vaccine and the race against the virus. Dive into the thrilling story behind the creation of the Oxford AstraZeneca vaccine and the incredible race against time in Vaxxers, the inside story of the Oxford AstraZeneca vaccine and the race against the virus by Sarah Gilbert. In this summary, you'll learn about the extensive research leading up to the vaccine's creation, the unprecedented speed of its production, and the challenges the team faced. Discover the crucial role that technology, collaboration, and dedication played in formulating the vaccine, as well as the continuous effort to overcome the evolving challenges of the COVID-19 virus, safeguard the world from future pandemics, and improve global cooperation.
Preparing for the next pandemic. Scientists have been researching viral outbreaks and developing vaccines for years before COVID-19 was ever identified. The book recounts the story of a researcher who encountered skepticism from a camper while discussing the lack of cell phone signal due to 5G towers. The camper revealed her mistrust of people making vaccines, which unknowingly included the researcher herself. The author presents the history of coronaviruses, their transmission to humans, and how previous outbreaks led to the development of effective containment responses. The book aims to educate readers about the importance of preparedness for future pandemics. Enhancing vaccine technology. Inadequate Ebola systems led to the development of a breakthrough vaccine technology platform that enables quick and efficient creation of vaccines for various diseases. The 2014 Ebola outbreak exposed the inadequacy of existing systems to combat swiftly spreading viruses, with no approved treatments or licensed vaccines available for the fatal disease. Although various Ebola vaccines were being developed, human trials were limited to 2 vaccines tested on rhesus macaque monkeys. By April 2015, the virus had already spread extensively throughout West Africa, and scientists could only test 1 vaccine at a time, due to fewer areas of high infection. However, the testing of VSV, an Ebola vaccine in one region with the help of contact tracing, quarantine, and ongoing clinical trials, led to the containment of the outbreak by June 2016. During this period, the ChAd3 vaccine was no longer necessary, leading to the development of ChAdOx1, a similar vaccine technology designed by Sarah and her team at the University of Oxford. ChAdOx1 is based on an adenovirus and is a replication-deficient, simian adenoviral vectored vaccine. This breakthrough platform technology allows for the creation of vaccines for various diseases, including those that do not yet exist, by creating a pre-designed framework with science-backed results. Improving virus-suppressing abilities through antibody production without causing illness is the essence of the technology that drives vaccine effectiveness and fast-track development of immunity against diseases. Overall, the Ebola vaccine paved the way for a vaccine platform technology that ensures quick responses and efficient development of vaccines against various diseases. The baker analogy of platform technology and vaccine development. The ChAdOx1 platform, likened to a baker who pre-bakes and decorates cakes, enabled rapid vaccine development. Professor Gilbert's team developed an influenza, MERS, and SARS-CoV-2 vaccine using the same platform, making vaccine development more efficient. With the MERS vaccine success, they already knew it was safe and effective before designing the COVID-19 vaccine. Less than 2 days after the virus's genetic sequence was made public, they had the vaccine's exact DNA sequence. The risky approach to accelerating vaccine development. In the early days of the COVID-19 pandemic, Sarah and Kath's teams at Oxford worked tirelessly to develop a vaccine by progressing through the different stages of development at risk. This method involved moving on to the next step before testing was finished on the previous one, greatly reducing the timeline but also risking wasted time and funding if unsuccessful. However, the potential benefit of saving lives outweighed the risk, and the approach paid off. Within 65 days from designing the vaccine, they were able to fill vials with the purified vaccine and begin scaling up production. Developing the Oxford AstraZeneca vaccine. overcoming production and accessibility challenges. The process of developing a vaccine involves not only ensuring its effectiveness but also its accessibility. The scientists at the CBF in Oxford needed partners to produce millions of vaccine doses. Working with AstraZeneca and others allowed them to secure funding, optimize production methods, and distribute the vaccine globally. Dr. Sandy Douglas developed a large-scale manufacturing method using 1,000-liter bioreactors to grow and purify millions of doses at once. The partnership also led to a vaccine that could be stored and transported at normal refrigeration temperatures, making it more accessible to parts of the world without ultra-cold storage facilities. The story behind the rigorous testing of COVID-19 vaccines. The development of the COVID-19 vaccine involved a rigorous testing process to ensure its safety and efficacy. It took 7 months to complete the efficacy trials. Scientists exercised caution and tested the vaccine on animals first before administering it to humans. The first round of human trials started with 1,000 volunteers to test if the vaccine induced a safe immune response in healthy adults. The Phase 2 trials expanded the age range of volunteers to include adults ages 56 and up without pre-existing conditions. Finally, Phase 3 was open to adults of any age and any health condition. The trials showed that receiving 2 doses 12 weeks apart produced the highest efficacy levels and reduced infection, transmission, and symptoms in cases of exposure. Preparing for disease. Why? With vaccinations being administered worldwide, preparations for the next pandemic must be made. This requires improvement in research and manufacturing infrastructure, public health response systems, and global cooperation. Disease. Why? The hypothetical future virus poses a great threat to the world's response systems. Industrial farming practices contribute to the spread of viruses, which can mutate and eventually infect humans. To ensure preparedness for disease. Why? Investment in improved vaccine development methods, stockpiling of important equipment like PPE, and ramping up of programs is vital. Funding and manufacturing infrastructure need to be in place to combat this virus, and the overall pandemic response will be more effective the next time around if these preparations are made.
In Vaxxers, Sarah Gilbert sheds light on the vital process of vaccine creation, its trials, and mass production that brought about the Oxford AstraZeneca vaccine. As the world grapples with skepticism, mutating variants, and global disparity in vaccine access, there's still much to be done in refining the overall pandemic response system for future outbreaks, such as disease. Why? By actively learning from the AstraZeneca experience, it's important to invest in better research and manufacturing infrastructure, enhance public health response systems, and engage in greater global cooperation for a more secure future against deadly diseases.